Peptides for transport into the endoplasmic reticulum are generated in the cytosol. Proteins in cells are continually being degraded and replaced with newly synthesized proteins. Much cytosolic protein degradation is carried out by a large multi-catalytic protease complex called the proteasome. The proteasome is a large cylindrical complex of some 28 subunits, arranged in 4 stacked rings, each of 7 subunits, and it has a hollow core lined by the active sites of the proteolytic subunits of the proteasome. Proteins to be degraded are introduced into the core of the proteasome and are there broken down into short peptides. Various lines of evidence implicate the proteasome in the production of peptide ligands for MHC class I molecules. For example, the proteasome takes part in the ubiquitin-dependent degradation pathway for cytosolic proteins, and experimentally tagging proteins with ubiquitin also results in more efficient presentation of their peptides by MHC class I molecules. Moreover, inhibitors of the proteolytic activity of the proteasome also inhibit antigen presentation by MHC class I molecules. Whether the proteasome is the only cytosolic protease capable of generating peptides for transport into the endoplasmic reticulum is not known. 2 subunits of the proteasome, called LMP2 and LMP7, are encoded within the MHC near the TAP1 and TAP2 genes. Along with MHC class I and TAP molecules, their expression is induced by interferons which are produced in response to viral infections. LMP2 and LMP7 substitute for 2 constitutively expressed subunits of the proteasome. A third subunit, MECL1, which is not encoded within the MHC, is also induced by interferons and also displaces a constitutive proteasome subunit. These 3 inducible subunits and their constitutive counterparts are thought to be the active proteases of the proteasome. The replacement of the constitutive components by their interferon-inducible counterparts seems to change the specificity of the proteasome. In interferon-treated cells, there is increased cleavage of polypeptides after hydrophobic and basic residues and reduced cleavage after acidic residues. This produces peptides with carboxy terminal residues that are preferred anchor residues for binding to most MHC class I molecules and are also the preferred structures for transport by TAP. MHC class I molecules also present peptides derived from membrane and secreted proteins. For example, the glycoproteins of viral envelopes. Membrane and secreted proteins are normally translocated into the lumen of the endoplasmic reticulum during their biosynthesis. Yet the peptides bound by MHC class I molecules bear evidence that such proteins are degraded in the cytosol. Asparagine-like carbohydrate moieties commonly present on membrane-bound or secreted proteins can be removed in the cytosol by an enzyme reaction that changes the asparagine residue into aspartic acid, and this diagnostic sequence change can be seen in some peptides presented by MHC class I molecules. It now appears that endoplasmic reticulum proteins can be returned to the cytosol by the same translocation system that transported them into the endoplasmic reticulum in the first place. This newly discovered mechanism, known as retrograde translocation, may be the normal mechanism by which proteins in the endoplasmic reticulum are turned over and by which misfolded proteins in the endoplasmic reticulum are removed and degraded. Once in the cytosol, the polypeptides are degraded by the proteasome. The resulting peptides can then be transported back into the lumen of the endoplasmic reticulum via TAP and loaded onto MHC class I molecules. And that's peptides for transport into the endoplasmic reticulum are generated in the cytosol. Have a great day.